All right, last part of protein synthesis. I'll tell you, in an ideal world, everything works as point part one, two, and three. So with transcription, translation, everything works perfectly. Proteins are made and life is good. However, we don't live in a perfect world. And we have what's called mutations. And the mutations occur where there is a change in the nucleotide sequence of DNA or messenger RNA that codes for a protein. Okay, so you can see they're very specific as to if we switch one of these letters, we're looking at a change, a mutation. So if we have a mutation in the piece of DNA and then that's transcribed, blah 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 blah, that's transcribed in messenger RNA, then that mutation is going to go all the way through. If there's a mutation in the messenger RNA, so we can have either one. So the messenger RNA that's going to code for the protein, it could come from the DNA or the messenger RNA. Now, these changes can be caused by many different things. And the cause, what's causing the mutation is called a mutagen. Remember, genesis is the origin or the start of something, and mute is for the mutation. So we're talking about to generate a mutation, to start a mutation. Examples of different types of mutagens: ultraviolet radiation, UV, radiation from the sun. So laying out in the sun, overexposure, people like mail carriers and people that work outside all day are at higher risk because they're exposed to the UV radiation so much. Cigarette smoke, huge mutagen. Alcohol in excess. Viruses, certain viruses can be mutagens. Car exhaust. So if you work near a place that there's an excessive amount of car exhaust, like in a auto mechanics shop or something like that, where you have to have proper ventilation, or chemicals can be mutagens. Things in the lab, pesticides, insecticides, different poisons, all of these can affect your DNA. All right, so there's two major types of mutations. We can have what's called point mutations or reading frame shift mutations. All right, so the point mutations are just that. They're at a point. A single nucleotide change happens in the sequence, and it affects a single codon. So you can have what's called a silent point mutation, where the mutation causes no change in the amino acid that's coded for it. We've never known it happens. There's no effect to it. Or we can have a missense. So here there's a one mutation, but it ultimately doesn't end up affecting the amino acid. We can have a missense where the mutation changes the amino acid that's coded for. So an A instead of a G, now it's going to change the amino acid. So this is a, quote, mistake. So when we see miss, think of it says like a mistake. This is best seen in the mutation that causes sickle cell anemia. And so from one change, obviously a dramatic, deathly, fatal effect. Or you can have what's called a nonsense point mutation. So all of these are point mutations. There's only one change. This mutation changes from coding for an amino acid to coding for a stop codon. So now instead of a mutation, so a U instead of an A will cause a stop codon instead of a particular amino acid. So no protein's going to be made in the case of a nonsense point mutation. So you can see there's no protein made. Miss, there's a mistake. So now we've made something different. And then of course the silent, there's no effect. All right. So let's look at the reading frame mutations. So this is where the whole DNA sentence is changed behind the mutation. So the whole thing, so nucleotides were added or deleted, and it affects the whole thing. So we can have what's called an insertion. And I need to point out that these reading shift mutations are alter the whole codon sequence. All right, so an insertion is adding a nucleotide. So if we add a nucleotide to the sequence, so an example of this would be as if we had, whoops, I'm not sure at all what has happened here. We just skipped back. Let's go to where we were. Okay, sorry. All right, so example, let's say our sentence is, the big tan dog ran. All right, so if we inserted the letters, if we inserted a letter, then suddenly it's like the boy, you know, I mean, you end up with this whole other sequence, and the sentence doesn't make sense. All right. So if we have a frame shift that causes a missense, a mistake. So here, this is now missing. And so you can see that now we've changed these two. So this is what was supposed to be. So methionine, lysine, phenylalanine, and glycine. Now we have methionine, lysine, and now we have a mistake. We have these two, and now the whole thing has shifted. We could have a frame shift causing an immediate nonsense. So a no protein. So now we have an extra U. So we've inserted something, and by inserting it, it causes it to stop. Or we can have an insertion or deletion of 3 nucleotides. And so we end up here, if these 3 were missing, then we would miss the lysine amino acid. And ultimately, if we don't have it in this order, it's a whole different, it codes for a whole different protein. And then the stop, so you can see if this is missing or if something is added in here. So we can have an insertion or a deletion for either one of these. Deletion, obvious, we're taking out a nucleotide from the sequence. And so you just make one change and it's a huge effect. So when we talk about gametes versus somatic cells, gametes are the sex cells. That's the sperm and the egg. You need to think in terms of that. So gamete are the sex cells. Somatic cells are all the other cells in the body. All the body cells are your somatic cells. So who's affected? If a mutation occurs in the somatic cells, the only one affected by the mutation is the person the mutation occurred in. If the mutation occurs in the gametes, the sex cells, the only one affected will be the organism created from the sex cell. So if the mutation is in the sex cell, in the gametes, if my gametes are affected, then it would be my children that would be affected. It doesn't affect me. But if there's a change in my somatic cells, then I'm affected, but no one else would be. So this is how future generations can be affected. And you have this change in the DNA over time. So if I do something, or if there's just a random change in my genetic DNA, then that can get passed on. Okay, that's it. Oh my gosh, we're done with protein synthesis. Have a great day.